In laboratories on Earth, viruses follow rules. They bind. They replicate. They mutate, slowly, predictably. But in space, those rules begin to dissolve. When scientists sent viruses beyond Earth's atmosphere, they expected damage, breakdown, failure. Viruses are not quite alive. They have no metabolism, no consciousness, no intent. Yet they evolve faster than almost anything we know. Instead, they witness transformation.
If you want to understand the raw mechanics of evolution, strip away complexity, viruses are the perfect system.
They are biological code, compressed, efficient, and brutally adaptive, which is why scientists ask the dangerous question: what happens to biological code when gravity disappears? Orbiting Earth is an environment unlike anything life evolved for. Microgravity alters fluid dynamics at the cellular level. Radiation penetrates DNA, introducing breaks and mutations. Electromagnetic fields fluctuate.
In space, cells don't experience mechanical stress the way they do on Earth. That alone changes how genes are expressed. Proteins fold differently. Molecular bonds behave differently. Even time at relativistic scales ticks differently. And viruses were exposed to all of it. When the first samples returned to Earth, nothing looked unusual. Until scientists looked closer.
We noticed altered virion structures, changes in symmetry and surface proteins.
Some viral capsids were more stable, others less rigid, but more flexible. Replication rates changed. Attachment mechanisms shifted. These were not random errors. They were adaptations. Whole genome sequencing revealed that both the bacteria and the phages on the ISS accumulated distinctive genetic mutations not observed in the samples on Earth. The space-based viruses accumulated specific mutations that boosted their ability to infect bacteria, as well as their ability to bind to bacterial receptors. Simultaneously, the E. coli developed mutations that protected against the phages' attacks, by tweaking their receptors, for instance, and enhanced their survival in microgravity. Then, the researchers used a technique called deep mutational scanning to examine the changes in the virus's receptor-binding proteins. They found that the adaptations driven by the unique cosmic environment may have practical applications back home. When the phages were transported back to Earth and tested, the space-adapted changes in their receptor-binding protein resulted in increased activity against E. coli strains that commonly cause urinary tract infections. These strains are typically resistant to the T7 phages. On Earth, evolution is constrained by ecosystems, competition, immune systems, environmental pressure. In space, many of those constraints vanish. When scientists sent bacteria-infecting viruses to the International Space Station, the microbes did not behave the same way they do on Earth. In microgravity, infections still occurred, but both viruses and bacteria evolved differently over time. Genetic changes emerged that altered how viruses attach to bacteria and how bacteria defend themselves. The findings could help improve phage therapies against drug-resistant infections.
It's like removing friction from a system. Evolution explores pathways it normally can't. Mutations that would be lethal on Earth persisted in orbit. Genetic diversity increased. Selection pressure became unfamiliar. Viruses began expressing genes differently. Not better, not worse, just differently. Some space-exposed viruses became less virulent. Others became harder to neutralize. A few interacted with host cells in entirely new ways. Interactions between phages, viruses that infect bacteria, and their hosts play an integral role in microbial ecosystems. Often described as being in an evolutionary arms race, bacteria can evolve defenses against phages, while phages develop new ways to thwart defenses. While virus-bacteria interactions have been studied extensively on Earth, microgravity conditions alter bacterial physiology and the physics of virus-bacteria collisions, disrupting typical interactions. However, few studies have explored the specifics of how phage bacteria dynamics differ in microgravity. To address that gap, Huss and colleagues compared two sets of bacterial E. coli samples infected with a phage known as T7, one set incubated on Earth and the other aboard the International Space Station.
We saw changes in host specificity, how viruses recognized and entered cells.
This raised unsettling possibilities. If viral behavior can shift this dramatically in space, what happens in long-duration spaceflight? What happens on Mars? Astronauts carry trillions of microbes with them, including dormant viruses, a closed environment, altered immunity, microgravity-driven evolution. Space doesn't just affect humans. It reshapes the biology we bring with us. Analysis of the space station samples showed that, after an initial delay, the T7 phage successfully infected the E. coli. However, whole genome sequencing revealed marked differences in both bacterial and viral genetic mutations between the Earth samples versus the microgravity samples. The space station phages gradually accumulated specific mutations that could boost phage infectivity or their ability to bind receptors on bacterial cells. Meanwhile, the space station E. coli accumulated mutations that could protect against phages and enhance survival success in near-weightless conditions. These experiments weren't designed to search for alien life, but they may have revealed something just as profound. Life might not need planets like ours, or chemistry like ours. When scientists sent viruses to space, they weren't creating monsters. They were uncovering a truth. Life is not fragile. It is experimental, restless, and always changing. And if something as simple as a virus can evolve beyond Earth's rules, what else might already be out there, evolving in ways we don't yet understand? Or evolution like ours? Viruses may be showing us how flexible life truly is.